Another screen grab of that tail.
Tilt down on the camera. All right, suction.
All right, suction coming up. Would you like it turned up all the way? We're at about 2200 right
Yeah,
now.
you can turn it off all the way.
Okay. All right.
So if you're watching us live on satellite feed 3, you can see the containers in which the slurp leads us to. It's sort of like a vacuum. Oh, there it is.
There it is.
Yeah,
It's inside.
success.
Very good. So, very nice. That's our sample 24. Thank you for that. Great.
Double check. I think that was jar number 2. Nope, number 3 we were in. Come down a little bit.
Roger.
Okay, so we are about 1,200 meters on the underwater, and we are slowly making our way up a seamount. I think the top of the seamount is going to be around 1,000 meters. So we're slowly making our way up there for the viewers that are tuning in.
Yep, that's right. We're heading to a summit depth of 1,170 meters or so, but we're going in a kind of clockwise spiral around the very top just to get a better glimpse of the different sides of this peak of the summit. So speaking of all of those cookie stars, I think next time we see one that is of slurpable size, we'll want to do a collection and a nice zoom. Just since we're seeing so many of them, they seem to be really characteristic of this region.
They all seem a little bit bigger than slurp size.
That's what I was concerned about. So if we could at least get some good zooms, and then if we happen to see one that's slurpable, we'll go for it. But
What's in the front box that we can't open?
yeah, what
Danger.
was it?
The front box has like an octopus egg and a squat lobster.
Okay, so it's the squat lobster who's our escape artist.
I think if we slurped it and then just open the box a little bit and drop the star in, we can do that.
Push it in there.
And
There's also a pink chrysovorid today, so I don't know if that is more floaty too.
It should be fine. They're generally a little more negative.
screen grabs. And all of those are in the port side box. So the starboard side box of the forward bio box is empty.
Okay.
So we could put it in there very carefully.
This one?
Sure.
Okay. Come wide, Aaron. Tilt down on the camera.
Okay.
So I'm kind of curious on the texture of the cookie star. Is it soft?
It's kind of in between. It's not super squishy or super hard. It kind of has sort of a scaly snake-like texture.
Ooh.
But on the bottom, it has the tube feet that you might be familiar with other sea stars. So that's what it uses to walk around. But it definitely kind of, really exudes that ravioli, the sort of firm but pliant outside and a squishy filling.
Inside?
This will be a grab.
Oh,
Yeah.
grab?
Oh,
No, not for you.
Oh.
Yeah.
Because we're putting it in the box, it gets classified as a grab, even though we're using the slurp.
The slurp is kind of a tool that you're grabbing instead of actually going into a chamber. But
I see.
this is
You
just
weren't-
for our
yeah.
own organizational
Back row,
purposes.
yeah, organization. All right, we can go for
Some back row things.
All right.
suction.
I just wanted to make sure.
No worries. Great question.
Yeah, I think you can use that little lip to kind of like nudge it up.
Ooh,
Ah,
there
there
it
we
is.
go.
Nice.
Oh, you got it up close to the bottom.
Yeah, could we get a zoom on that?
All right, push in there.
Wow.
Nice. That's an excellent image. Yeah, get some good screenshots of that. Fantastic.
So that's what would open,
Okay,
and then they
my
would
God.
cover the coral.
Yeah, so
Rack back the camera
in
a bit.
the middle is its mouth, and it would actually just extrude its stomach out and wrap around what it wants to eat.
Wow.
All right.
Kind of scary and interesting.
Yeah, it's a little creepy, but in like a really fun way.
Yeah.
So yeah, if we could go in that starboard side of the forward box.
Okay. All right, if you want to pump out the box a little bit.
All right. Pumping out. Oh, that's quite a bit.
All right, can I? 1 second. All right, kill suction.
Stop.
Perfect.
All right, excellent. Closing.
All right. Success.
Nice.
So you guys mentioned there's an octopus egg in one of the storages?
And sometimes the deep sea octopuses will lay their eggs on chrysogorgic corals.
Huh.
So they,
Yeah.
not all the
Tip.
octopuses brood their eggs. Um, the cirrate octopuses will actually lay an egg sac and leave it.
I see.
And those are the octopuses with the flappy ears.
Ah. So I think, I guess like one of the comments from the viewers are asking how it would, like the chances of it to survive when we bring it up, which is probably not likely, right?
No, the change in temperature
Pressure.
from the deep sea to the surface is usually really stressful for these animals, so they don't come back alive.
Yeah. But I think it gives us an opportunity to study them and get, yeah, so it is also important, so, yeah.
Okay.
We're going to do a little pilot swap. Auto heading on, and we've just been, we've been crabbing along kind of to the south along the ridge, so
Yeah, my last move was due south.
just barely coming up slope a little bit. Yeah, so you kind of see like the nav screen, like we're out a little bit ahead of Atalanta, and they're both the vehicles are kind of moving south.
So as we see more of these cookie stars, we could just get a couple of zooms on different individuals, not collections, but just zooms to see if they're all the same, or if we're seeing some different species. That would be great.
Oh, that's different. Not a cookie star.
Awesome, yeah. This is what we're trying to figure out. If all these white cookie star-like,
Oh, maybe it is a cookie star. It's just really weird shape.
it's just eating.
Yeah, I thought it was something else.
Can we zoom any more?
We can, but it'll go off screen if I
I
Okay.
think the camera needs to be moved.
Tilt up.
Thank you.
And this is the same as what we collected. It just, it's got some weird posture going on.
Just different
Made it
shapes
look
and
like a different.
sizes.
Here, I'll just zoom out a little bit. There you go.
Yeah, all or nothing. Okay, I think we're good then. Thanks.
Cool.
Great. Thank you.
Mm-hmm.
So there's not much coral around. Do they also eat like sponges and stuff?
Generally they don't eat sponges.
Mm.
Sponges are mostly glass. They're hard to eat.
Oh,
But they could be feeding on other things for sure.
okay.
Okay, yeah, that's good.
That's great. Thank you.
So I'm sorry, this is a different from the ravioli star, or are
Oh,
they
that's the same.
okay?
These are all the same.
Is that an anemone growing on a glass sponge?
Yep, that's that Actinoscyphia Venus flytrap anemone
Nice.
on a Regadrella glass sponge.
Can we get a quick snap zoom on that? Nice. Beautiful.
Is that a little coral in the background?
It is.
So, is the lattice structure that we see in on
It's great.
right
Thank you.
there the spicules?
Yeah, it's all spicules. And there's tissue stretched in between those spicules.
To make that pattern.
Yeah. Yeah, so like the spicules make the pattern, and the animal itself is like that. The spicules are the skeletal structure, and then the tissue overlays it.
Okay.
So shrimp. Those are some dead sponges, right? We're seeing.
Yeah, the brown is the dead sponge, and then that when it's alive, it's that sort of almost bluish white color.
Yeah, we saw one of those earlier.
The
Two of them.
conolasma.
Yeah,
Yeah, it's super crunchy.
another one.
So May, this is pretty much similar to our last dive, right? With the way the rocks are, it's mostly volcanic.
Yeah, so we're on a seamount quite nearby our previous dive. The difference with this seamount is that it's a little bit deeper, so its summit depth is a bit deeper. And we were having some debates about the last dive's geological structure being carbonate or basalt at some parts of the dive, but in general, all of these seamounts were formed volcanically, so they're made of basalt, and they're quite old, so we see this manganese oxide crusting and sediment dusting on top of everything. But in the shallower depths of the seamounts, we'll have this carbonate structure forming, which is from old ancient reef structure, biogenic carbonate.
Interesting, thank you.
That's definitely part of the objectives of this dive and throughout this expedition to,
You want to zoom that in, enemy?
yeah, let's check it out. To characterize the geologic setting that all of this deep ocean biodiversity and abundance is living on and around. Little brittle star. Oh, is there something growing on that? Or is that just kind of,
I don't know.
you see
It
that?
looks weird.
This little,
It's growing an arm.
yeah.
Yeah.
Is it a parasite? Can't really tell.
That's all the zoom I have.
It looks like it's part of the anemone.
Yeah, it's like the same color, same fleshy material. Maybe it had some sort of,
Yeah, maybe
it
there
grew
was
around
some,
some sort of,
yeah, some
like,
damage or something, and it
exactly, yeah.
grew a little funny. There's a small bamboo coral too, and a brittle star.
Yep,
All right, thank you.
great. So there was sponge.
Okay, so someone's been paying attention. One of the viewers is asking, earlier you said sponges are mostly glass, then describe them as crunchy. Could you please elaborate? Maybe there's a question for Megan.
um, well, we kind of describe them as being crunchy or squishy as a way to better understand how to collect the specimen. Some sponges have spicules that are arrayed in a tighter lattice work that doesn't flex and bend very easily, and so those are our crunchy sponges. Those tend to be in the families Yurrididae and Farreidae. And then we have more of the squishy sponges that have these longer, more flexible spicules, and so they tend to be a little more pliant when touched, and those are the sponges in the family Rosellidae and Euplectellidae. And so they'll squish and spring back into shape instead of fracture and crunch like chips.
Thank you, thank you.
Oh, there is a fish.
Yeah, let's see if we can get a zoom on it.
There's two fish. There's
Ooh,
one
there's a big fish.
Go big fish first.
Yeah,
big
big,
fish.
big fish. Big fish, little fish.
I'm going to take off auto heading and let you chase it a bit.
There's a stalked sponge off to the port. More of those cookie stars.
So this is a cusk eel. It's got a very narrow, long head.
give it some extra beans.
Yeah,
Turn
it's
around,
swimming
bro.
uphill.
Smile for camera. Mm.
I'm going to zoom out.
Nice. Get some good images of that. Awesome. We may have lost our little fish.
Yeah, we went kind of far away. But I think if you just back up.
Yep, you gotta back down a bit. Auto heading back on.
Oh,
Jellyfish or something.
Jellyfish.
Okay. Back in the box. Want to send a DVL reset, Megan?
yeah.
10 meters off.
Alright, and if you're just joining us, we are about 1,200 meters down in the ocean.
Have to fly with it.
Mhm. There.
No worries. We've got plenty of time.
So it's natural. One of the things you have to fight is when video zooms in, it's natural for the vehicle to start drifting back, even if you're not giving it a command. It's just like human instinct. So you're backing up. So you have to, like, if you watch the downward cam, you can try to fight that and like make sure you're giving some stick forward and downward as the video zooms in, so that you're not drifting back from it. Because once you get further away from the further away you are when it's zoomed, it's harder to maintain it. And you want to be as close as possible to the item that you're zooming on.
Oh yeah.
Is that a
This is a rat tail fish.
I wouldn't land with the fish. I would try to see how it's drifting with it. I would just try to drift with it. You got it. Nice.
Oh.
This is Coryphonoides.
Oh. There's a little beard.
Jeff agrees.
No, we haven't seen this one yet today.
Oh. So it's not the rat tail?
It is a rat tail.
Oh, it is a rat tail.
But this is a Coryphonoides,
Oh.
so different genus than what
I
we've
see.
been seeing before.
Rat tail is just sort of a general common name for a lot of these fish.
Yeah. Yeah, so
Has a really
The
long.
family Macurinae includes all of the rat tails, but there are lots of different genera within that family.
Wow.
So our one
So
of our
funny.
lead scientists and watch lead on the previous watch, Jeff, he said that they saw one on the previous watch actually,
Oh, okay.
but just
Wow.
one. So we've seen a lot of nice fish on this dive. It's interesting going through the crinoid zone and then into the cookie star zone.
Yeah.
So is grenadier another name for the rat tail?
Yes, it
Yes.
is.
Okay.
It's the name they use when they cook it.
Yeah, I think I remember.
Yeah,
Uh,
rebranded.
It's the rebranded.
Yeah.
Yeah, we can let this fish go.
Great, thank you.
Oh, that sponge is slowly dying. It looks like it changed colors, changing colors.
Just stretch all the way out. You're gonna have to kind of lateral left and back.
Still seeing a lot of those white cookie stars.
That's the same fish.
Mm.
Mm-hmm. You can tell it's got the split in its pectoral fin there.
He's like, you didn't get my good side. So if you are just joining us, we are about 1,222 meters down on the seafloor. There has been no previous dives that has been conducted on this unnamed seamount, which was mapped for the first time by EV Nautilus this morning. So our team, including Megan, they worked really hard to map this out, and yeah, here we are trying to explore, trying to find things that see the diversity and the distribution of the deep sea communities. Try to see what types of fish is here as well, as we slowly make our way up to the top of the seamount.
Yeah, you're right. Bamboo. Good ID.
This one's really susical.
Mm-hmm. Especially with the Actinoscyphia in the middle there.
Oh, and a fish.
Oh, yeah.
There he is.
It's presenting itself. So is it, Megan, I don't know if you would know, but we can see that the polyps along the bamboo coral sort of stop where the anemone is.
Mm-hmm. So usually there might be some damage to the tissue and the anemone settled in that area.
Okay.
Generally, they don't overgrow live tissue, but it could have, as it's been growing, pushed and competed with the bamboo coral polyps themselves to make a lot of room for itself. So there could be some competition there.
Okay.
So we have a question. Maybe we could answer this. What has been the coolest thing we've seen so far on this watch?
Ooh. I bet everybody has their own answer to that question.
Yeah.
Yeah,
Yeah,
that
the
was
slick head.
that was the highlight, the slick head fish.
What kind of fish was that?
Were you in, you maybe were
missed
in the
it,
interaction, yeah. Yeah, we saw a really, really big fish, a slick head fish, right?
Yep.
And it was well over a meter in size and probably 25 centimeters wide or something.
Jake did some amazing flying, and it was very cooperative.
It looked massive compared to all the other fish we've seen on this dive and this expedition. So it was quite cool.
Yeah, just waited for me to get out of the room. Huh?
I'm sorry.
It always happens.
Sometimes it happens like that, but it was in the background of your interaction. So
I'm sure the kids really liked it. They can see it behind you.
So.
they can see the screens behind you.
Yeah, so I've done a couple of interactions, and it's sort of funny how you're oblivious to what's happening behind you, and then it'll be like in the background, like an amazing thing happening.
Exactly. It's part of the excitement of doing this work is we really don't know exactly what we're going to see or when.
Until you see it.
Yeah, we have some general expectations based on the bathymetry or the seafloor features and the depths, but the rest, that's what we're here to do. We're here to map these habitats and understand the life and geological setting.
Yeah, just see whatever the ocean will offer us in
Exactly.
this little window of time.
Exactly. Yeah. There's so much that we'll certainly miss, but we try to plan a track of the features that seems like perhaps the most interesting one, and then we adapt those plans based on how things are going. So right now we're kind of doing a little clockwise lateral motion around the summit since we have a bit of extra time, which was originally unplanned. And that lets us see what's just out of view from our original path and follow some of the contours as we circle around the seamount
Oh, that's a rock.
and get a look at a different side of the seamount. Since some of the interests of our scientists are kind of the intersection of geologic features, current regimes, and how that affects the biogeographic settings of this region. So we were looking at up a different side of the seamount slope yesterday. We were more on the northwestern slope, and now we're kind of taking a clockwise turn, going south and from where we started to sort of see a different angle of the seamount of this particular one.
All right.
See how the currents affect this side.
How many interactions have you done so far? Because it's only a couple days in.
I think 4 to 5,
Wow.
maybe 6.
That's pretty cool.
Yeah.
It's a lot.
That's a lot of little kids who are getting to see.
Yeah, it was. It's been really good trying to talk about it, and I think the more I'm doing it, the more comfortable I am with just trying to operate. When you helped me out last night, I was like panicking. I didn't know which button to push.
Listen, it's a lot of buttons in
Yeah.
there.
But it's getting better.
No, you did great.
Thank you. Yeah, you helped me a lot.
It's just already impressive. We're only a couple days in and you've already got quite a few.
Yeah.
And it does make a big difference. We do them to my mom's class when I'm here.
Yeah.
And there's some kids who've never heard of deep sea ocean exploration, and suddenly that's what they want to do.
Yeah, I'm excited. Hopefully we get that rescheduled.
Yeah. Especially if they see someone from where they grew up doing it,
Yeah.
it suddenly becomes a lot more possible.
Exactly. Yeah, I think the kids saw some of my relatives, so they were trying to wave to me while we were doing the previous one.
I bet they were really proud.
Yeah. And we kind of did it in Palawan, so the kids were excited.
How special is that? That's really cool.
Nice shot.
All right.
Nice, Sarah.
Very beautiful.
Hello.
I'm going to go in just a bit more.
So based on its kind of naked snout, it looks like it could be a Nizumia, is what our fish expert Jeff on board is suggesting.
I'm going to zoom out a bit. There we go. I'm sure someone was wondering.
We have a question from the chat if we've seen any hydrothermal. We have not, right?
Seen any what?
Hydrothermal vents.
Ah, no,
Yeah.
not in this area.
So this is most likely inactive.
Yeah,
Yeah.
yeah, these are really old volcanoes, and what we're more interested in is looking at the kind of ancient life growing here, and all life for that matter, and dating these ancient volcanoes as well.
I think in Canada, when they did, it's the Canada where they found a lot of the thermal vents.
Yeah, definitely,
Yeah.
and also near American
Too
Samoa, and
close.
next year there will be some work
No
near
good.
hydrothermal vents near Fiji,
There we
and
go.
Yeah, a lot of organisms thrive there, right? Because of the...
definitely, yeah. The hydrothermal vents are more of kind of what my background is in, and they can be very vibrant environments as well. Different source of food, different source of energy, and as I really like how Daniel Wagner puts it, as the kind of ancient sort of old growth forests of the deep sea are these kind of coral and sponge communities that grow on these old volcanoes.
Is there a chance that they would turn into volcanoes?
These ones not.
I meant the thermal, the ones you've seen, that's coming out with a lot of heat coming out of the. Do they?
Yeah, yeah. So the areas that are volcanically active can be erupting lava on the seafloor, creating seamounts, or at tectonic plate boundaries, a process known as seafloor spreading occurs, where new seafloor is made as the plates are spreading apart. And elsewhere on the planet, the plates are subducting under each other. So in total, the planet isn't growing or shrinking in size. It's all kind of this big puzzle of plates moving around and shifting. And those volcanically active spots can still remain hot, even if they're not erupting magma or lava at the time. And just as gravity brings things down, seawater will percolate through the cracks of the seafloor and get warmed up from that volcanic heat source and dissolve all these minerals from the crust and emit back out and form a kind of chemical-rich energy source for life.
To thrive.
Yeah.
Interesting.
But yeah, if you are interested in learning more, we've got lots of videos of previous expeditions that looked at hydrothermal vents on our YouTube page, and we're also going to be visiting some next year, most likely.
Look at that jelly.
Oh,
He's trying to fly away.
it's kind of cute.
Yeah.
Very decorative.
I think it's gone. Oh. Just kidding.
Have you guys had a chance to go to Jellyfish Lake yet?
No. I
No.
want.
Okay.
Yeah.
Maybe after the
Hopefully.
cruise.
I think some people are staying around after, and perhaps we'll visit. It's definitely world famous among ocean lovers, that's for sure. Do you want to share a little bit about what that is for
Sure.
For the viewers, yes,
the viewers?
please.
Yeah, so one of the really popular tourist destinations in Palau is called Jellyfish Lake. So this is a lake located in the Rock Islands, which is another really popular tourist destination. It's a marine lake. So it's enclosed on all sides, but it has vents which allowed seawater to come in. The species that is in that lake is called Mastigius papua etpisoni, which is actually named after one of our, speaking of naming species and seamounts, is named after our former president, Niratkel Epison. So
Wow.
this is a subspecies that's found only in Jellyfish Lake because it's been entrapped and evolved separately from other species. So they are stingless, so you are able to actually snorkel in the lake among all of the jellyfish, and you will not get stung by them. Other interesting facts about the lake is that below about 13 to 15 meters is toxic gas, so no diving, only snorkeling at the surface. And I've had the opportunity to go to the lake a couple of times in my life. Definitely an otherworldly experience, but it is sort of sensory, having a lot of the jellyfish touching you at the same time, so it can get some taking used to. Yeah.
It's really neat.
Yeah, learning something. I didn't even know about the name. Appreciate you for sharing.
Oh,
Yeah.
about the scientific name.
Yeah, how it was named after a president.
Yeah.
It's really
Cool
cool.
stuff.
The common name is golden jellyfish.
Golden jellyfish.
And there actually are two species. There's a golden jellyfish and then a moon jellyfish, which are a lot smaller, and they look pretty similar to the ones we've been seeing on the dive.
And they also don't sting, yeah?
That I'm not too sure of. Maybe they do, but they're pretty small and tend to stay away from humans.
It's really cool. I didn't know about the toxic gas.
The toxic layer.
Yeah.
It's not a part of the highlight reel for sure.
Yeah, I guess that's not advertised.
But when you go to the lake, they do
Advise
inform you so you don't try
Right.
and dive to the bottom of the
Sure.
lake.
That would make sense.
Or free dive. Yeah. 30 meters. Yeah, so the
Okay.
top 13 meters are good to go. Below that is not good to go. Yeah, and it's a little hike through the rock island, so
Goes
to get
up,
there is a
yep.
bit of a hike.
Goes up and then it goes back down.
Cool.
Yeah, I've also been there a couple times.
Nice.
It's such a good experience.
It's really cool.
But there was a time where the population kind of declined.
Okay.
I don't know if that was due to heat or something. I can't really remember, but yeah, I think it's done a really good job recovering back. So
Oh, nice.
Yeah, so the population of the species in the lake sort of dropped around 2016 and 2015 to 2016, which just so happened to coincide with a tourism boom in Palau. So there was some talk that it was due to that, but after some studies by the Coral Reef Research Foundation, they think that it has more to do with the temperature of the lake.
Okay.
Yeah.
Interesting. Just a shout out to our previous ocean science intern who was on Lebu's Voyage 1, our first expedition. Ayla, she works for the Coral Reef Research Foundation and often goes swimming in these lakes and studying these populations of jellyfish and spending time in these saltwater lake environments around Palau.
Yeah, so Jellyfish Lake is actually one of many marine lakes that are located in the Rock Islands. And yeah, CRRF is doing a really good job of cataloging and studying all of them. This one, I believe, is the only one that's open to the public, and the rest are sort of, if you're adventurous, I think you can get to them, or if you're a scientist, as scientists tend to do, go to the less explored places. And yeah, I think they've seen like crocodiles and a lot of less friendly than jellyfish.
That's what Ayla was sharing with us before.
Yeah.
Even just walking on land, you can run into crocodiles as well as in the water, heading out to these more remote marine lakes
Yeah.
to monitor them. It's really important work that involves a kind of adventurous sort of spirit.
Good stuff.
Yes, and Palau is actually the only country in Micronesia that has crocodiles. So,
Really?
yes.
Wow.
We
I
have
didn't
a
even know that.
saltwater crocodile named Crocodylus crocodylus, and that only occurs in Palau.
Wow.
Yeah, out of Micronesia, which is, for those listening, the Pacific region is sort of generally divided into three. So Micronesia is Palau on the sort of the northwestern Pacific.
Small islands, micro.
Small islands, yes. The middle, which is consisting of maybe the better known Pacific islands like Samoa, Tonga, sometimes Fiji is Polynesia. Yeah, Fiji sort of goes in between categories. And then Melanesia, which is sort of to the south, so
PNG.
Vanuatu, PNG.
The darker, yeah.
Yeah, so Polynesia means many islands, Micronesia means small islands, and Melanesia is, as you can guess from melanin, dark islands. Oh, a fish.
Yeah, that was another rat tail, probably the same one that you were looking at earlier. Yeah, we don't need to chase down every fish, but it's nice to get more fish than, you know, on previous cruises. Sometimes we get distracted by the sessile organisms and don't census these more modal organisms first. So a good balance between looking at fish and looking at corals and sponges is always good. Sure, let's do it. This is a glass sponge in the family Euplectellidae called Ragadrella. Yeah, these are the ones that the shrimp couples inside.
Structure is just so amazing how it grows.
Yeah, it's really beautiful. Very delicate.
Yeah, so the water flows through the sponge, and then the sponge can pick up
Absorbs.
all the little food particles that are in the water. So it's really important for a sponge to have lots of holes and ways to move water through its body in order to feed.
And growing up vertically into the flow of the current definitely helps with having more surface area and more room to get those little
Catch.
particles. Yeah. So you'll see that often they'll be wider at the top than the base.
Mm. Some more starfish.
Lots of them, yeah. Were you here for our collection of one of those?
Yeah.
Yeah, cool.
Oh, what's that red? Shrimp?
Another anemone. Sure.
Sure.
Let's do a zoom.
There's that red shrimp. It's floating by. Oh, another one. It's like chilling. Going with the current.
Oh, there's some bryozoans too. Those are those little floaty feather-like structures.
Oh yeah. Over here.
Mm-hmm.
Is this some kind of encrusting sponge?
Yeah, that is an encrusting dema sponge.
Nice. We've seen a couple of those earlier.
Mm-hmm.
Yeah, right over here. A lot going on when we take a zoom.
Oh yeah, for sure.
Nice.
So you said encrusted sponge. So does that mean it's just growing with?
Yeah, it just
rock?
grows like a thin layer over the rock.
Okay.
Instead of growing its own sort of structure or skeletal structure, it's kind of,
It's encrusted.
yeah, it's using the structures that are already there.
But essentially it does the same thing of absorbing micronutrients.
Mm-hmm.
Yep, exactly.
He's a renter, not a homeowner.
We got about 30 minutes left on our shift.
That's right.
Hopefully we find something.
Do we have anybody tuning in from Palau, Bloda?
Yeah, we've had some viewers from Palau so far. Yeah, we got some from the States. We got 6 from Palau that's watching. We got some from Australia, Canada, Russian Federation. We got some from Philippines, shout out. Yeah, New Zealand, Republic of Korea, wow. India, Ireland, Guam, United
Yeah,
Kingdom,
let's go for it if
Czech
you can.
Republic. Welcome all
I think
viewers.
it's a cyclothony.
Awesome.
Is this a very small fish?
Yeah, this is a hard one to zoom in on.
It's really fast.
Wriggly.
I'm ready.
Gives her a challenge.
Here's a crazy question. Do rising ocean temperatures affect the depths as much as the surface layers?
Yeah, well,
I think it's a mix. Oh, there.
all of these waters are,
I'm going to zoom out. It was a good effort.
Did we lose the little fish? Yeah, no worries. It was a very fast, wiggly one.
Too fast. Too furious.
Yeah, definitely the rising ocean temperatures are going to have a whole bunch of cascading effects across the oceans. Certainly they will be felt quite strongly in the shallow waters and will be affecting coral reefs in shallow water settings as they have been already quite significantly. But these waters are all interconnected, and currents and the flow of ocean waters are influenced a lot by temperature as well as winds, and those cycles will be altered and potentially balances will be changed and currents may be changed as well. So often these kinds of flows are responsible for carrying food sources, so you can imagine how that will also affect the deep ocean. Partly why we're so interested in this region is that it has a combination of factors that leads to tremendous biodiversity and abundance in the shallow water setting. So we're interested in how that looks in a deep water setting, given the convergence of several current regimes and these interesting features, these seamounts and ridges that provide habitat. So the short answer is yes, but
We love a long answer.
but it will be affected in different places to different degrees and on different time scales.
Because everything's all connected, right? So, of course, if something changes here, it's going to make some changes there. Maybe minute, but eventually some changes. More things. I appreciate that.
Yeah, and these deep ocean settings are really quite ancient, slow-going environments, so they're not accustomed to the rapid, accelerated kinds of change that are human-induced, or even change of that nature that might be more quick to happen in surface or terrestrial environments. So it's another added layer to it, that fast changes are also something that will be difficult for these environments in the deep ocean to adapt to. So another importance for preservation and conservation, and Palau is quite famous for having such a massive amount of its waters preserved. 80% of its waters are part of the Palau National Marine Sanctuary. And a big part of this expedition and the previous one is characterizing the deep ocean habitats of those waters to help with conservation efforts with our partners at Pickrick and the National Marine Sanctuary.
Yeah, thanks, May. And something really interesting as well is that the 80% is actually only counting 80% of Palau's exclusive economic zone in our national waters. And it's really, I'm really happy to add that in our shallow waters, which is the first 12 nautical miles, we actually have 30% of our ocean protected as well with the
marine protected areas.
Marine protected areas with a scheme we call the Protected Areas Network. And that is a part of Palau's commitment to a worldwide effort to protect a certain percentage of the world's ocean in order to help it absorb those changes from climate change. So Palau is doing really great on that indicator, and all of the efforts to do research within those protected areas, of course, will lend to the success, the long-term success of keeping them protected.
It's fantastic.
Yes, thank you for adding on to that.
Yeah.
Gonna
to say at Atlanta's, I say that wrong. The view on sat feed 2 is very cool right now. You can see Herc pretty well.
Yeah.
It's a great point, yeah.
So we can usually do this when the currents aren't too bad, right?
What's that?
We can usually do this when the current isn't too bad.
Like have the view like that?
Yeah. Because usually we're all the way on top.
Yeah, it depends on how the tether's laying out. So yeah, so it does have to do with the current. So like the current's kind of pushing the tether back, but now that
looks really cool.
when the vehicles are kind of on top of each other, you gotta raise the delta a bit to keep the tether happy and not tangled up.
All right.
So as
Thank
Atlanta
you,
comes
Jake.
on top right now.
This is a glass sponge called Trita pleura.
Trita pleura.
Mm-hmm.
Wow.
That's good, Sarah. Thank you.
Yep.
Nice.
Sarah, you're rocking it.
Yeah. Really great flying.
Couple of really long, are these also with, bamboo coral?
Yes, these are bamboo coral.
Wow, those are probably really tall.
Definitely.
Are whip coral and bamboo coral, they're different, right?
No, it's kind of like a colloquial name for it. Yeah.
Whip, whip
Yeah.
coral. But the common name is bamboo.
I guess so, yeah. I guess that would be considered a common name.
Okay.
Yeah. Yeah, we collected a nice one yesterday, which was quite long and wiry.
That was the one that branched?
Remember, Taya? Yeah, we processed that, the branching one.
Oh, cool.
Took us a long time to measure.
Oh my goodness.
We miscalculated. We needed about 10 centimeters and ended up getting quite a bit more, but
Oh, how much did it end up being?
I want to say it was like 75.
Wow.
Well, it's hard to tell. With
our
the,
scale was off.
Yeah.
And I guess it was so large that that small snip did not appear to be
A small snip.
a small snip.
Yeah, so these bamboo corals can get really, really long then, I guess.
Definitely. I keep asking Megan this, but do you know the growth rate for bamboo corals?
Yeah, bamboo corals, there have been some studies on them, and you can measure their growth rate by measuring the width of the base. So the width of the base will increase in a linear fashion. And so by measuring that, you can estimate how old it is. So something with a pretty decent sized base can easily be 100 years old. So like those long whip corals are over 100 years old.
Oh, it's fascinating.
This is a really cool sponge.
Yeah, it's really beautiful.
It's a sponge, right?
Yes,
Yes.
this is a
This is
glass sponge in the family Euplectellidae
Crunchy?
called, it's soft and squishy.
Ooh.
Yeah.
Squishy.
Yeah, you see how that matrix is kind of very mesh-like, like the regadrella. That indicates that it's squishy. It's got these long spicules that'll be very flexible. This sponge is called Sacacalix.
So cool.
It looks very flower-like, very
Yeah.
beautiful.
I think they would make really great lamps, you know, like for above your
Oh.
kitchen, you know,
Yeah.
like pendant lights.
Get IKEA on the
Do a
line.
3D model and then put a light.
That would be cool.
Chandelier.
Could we 3D? I don't know. That seems complex to 3D print.
Hey, they're
Be
3D printing
surprised with,
everything, yeah.
yeah. I think it would be more beautiful to do some glass work.
Oh, yeah, even better, right?
Make a glass sponge out of glass.
Exactly, yeah.
Oh, that would be a really awesome art project.
I've done some glass work with lava, remelting lava into some glass forms, which was really
That sounds
fun.
beautiful. Is there any way
Ooh.
we can see? Ooh, jelly.
Hello.
Ooh,
It's a Perifragella.
Oh,
such delicate little, nice.
Oh, the way it swims.
This look like the top of a beetle nut,
Yeah.
swimming or a coconut, yeah.
So in Palau, we have a tree nut called, well, the common name is beetle nut. And you would chew it, and it gives you sort of like a high. I don't, personally, I don't chew. I don't know.
Me neither.
Is that the one that makes your mouth red?
Yes. Yeah.
It's pretty common. But it's, yeah. Ooh.
Ooh.
Turn around. Yeah.
The conical shape.
Yeah, it's really beautiful.
That's a really nice
Ooh,
close-up.
that's a good zoom.
Can we turn the lasers off?
Lasers up.
I wonder if the conical shape of its bell is to help its sort of hydrodynamic movement, or if that's not really a factor in bell shape.
I don't think that's really a factor for this one.
It's not trying to go zooming around, I
It
guess.
doesn't zoom very well, no.
It's cool with the cookie in the background.
That's about as fast as it goes.
It flutters.
Yeah.
What do you think impacts the shape of the bell from being?
I think it's based on species. Different species have different shapes. This is a coronate jelly.
Do you think there's any sort of benefit, or it just happens to take on that shape?
I'm not sure. I don't know that much about jellies.
A lot of glass sponges in the background.
Jeff is saying Periphylla.
Yeah.
Thanks,
Nice.
Jeff.
Atalanta's getting ahead of you.
It's a very beautiful way to end our watch.
Nice work.
Good job, Sarah.
Thanks, Sarah.
Wonderful flying. Our pilot switched a little while ago, so fantastic.
So to our viewers, we are still about 1,200 meters down. We are slowly going around the seamount, making our way to the top. Yeah, so hope you guys stay tuned with the next watch that's coming in. They should be finding some interesting things on top of the seamount, hopefully.
Yeah. And so we'll have probably another 3 hours or so bottom time. And as we make our way up to the top, we'll keep an eye out for any other interesting species to zoom on. I think we might try to, maybe not, depending on space, we might try to collect a rock at the top, and then on our way up, we'll do some eDNA water sampling and keep moving along, and we'll be diving for the next several days here. So a lot more exciting seamounts and features to dive on.
So this looks like it's not really going to go as much higher than what we are right now, right? Just a couple more meters.
Yeah, so the summit depth is around 1,170 meters.
So about another 100 meters to go.
Yeah, but as we're kind of reaching our summit, we're kind of taking a clockwise
Around it.
spiral around the top just to get a lateral view and see if there's one particular direction or angle of the summit that seems to be flourishing more than the others based on the currents. Yeah.
Based
Cool
on
stuff,
other summits that, in general, that we've seen, I've actually never been on a watch where we summited. Could we expect to see anything in particular, or just because of how the current might be at the summit?
On the summit? Yeah, the summits are really exciting. Often they're well positioned in the currents, and we can often see quite abundance and diversity of life at the summit. So it really depends on the depth and the current regime, of course. It's not always the case, but I believe our first dive and our last dive of the first expedition, we had some spectacular summits with really, really exciting coral and sponge habitats with numerous species and different colors and a lot of abundance that we saw really booming at the top compared to the flanks, where it was still quite biodiverse, but not like what we were seeing at the top.
Cool stuff.
See more cookie stars scattered all over.
Yeah.
Jason said he would try the beetle nut. Just like he heard about the nut. He won't lie, he would try it.
Yeah, I mean, when I say I don't chew, I mean I have tried it. But there are people who are regular chewers, so they have a bag that they regularly fill with the beetle nut, and they tend to say they need to chew after they finish eating. Just because maybe if they feel full, it helps them with that fullness. Yeah, and it gets you you get a little feeling from it.
I've never tried. I think I was traumatized as a kid. Yeah, so I've never tried.
Is there a cultural kind of layer to it as well, or is it more just sort of a activity?
No, there's definitely tied to culture. You know, back in the days, the men would come together and have a problem and kind of chew it out and discuss what's going on. But yeah, it's always a good time for them to kind of pause and think and chew and think of their words before they. Yeah, so to me, I don't know. I couldn't. Like I said, I think I was traumatized because my older sister kind of forced one on me, and that was just like they held me down. My brothers held me down, and my sister was just trying to stick it in my mouth. I was like, yeah.
Oh no.
I'm kind of glad though, kind of.
So that you didn't grow up a chewer?
Yeah, I didn't grow up a chewer.
Yeah.
Is that a dead coral?
No, it's alive.
Oh.
We're gonna take a look at it.
Yeah, let's zoom on it.
Ooh, it's nice and flimsy.
It's wiggling.
So does that make it a soft coral?
This is likely Paragorgia. Have we collected anything like this on this cruise or last cruise?
On this cruise or on this dive?
Oh no, on either cruise have we collected a white coral like
white
this?
white Paragorgia? Yeah.
Okay.
Yeah, we did. Last cruise. But we'd have to zoom to see a little bit closer, but we did collect a white Paragorgia. Had a different branching pattern, so I'm not sure it's the same, but its texture looked quite similar.
Is it wiggling because the fans of our Hercules?
Yeah.
Okay.
Thrusters.
The thrusters, yeah.
Yeah. Causing a little microcurrent.
Oh, wow.
It's a squat lobster. Nice.
There's a couple of...
Well, it's just a little one.
Yeah, these look like amphipods. This
associates.
rhizome. I'm gonna try to take a still. See how it goes, since we're not moving.
So to our viewers, we're slowly getting in the next watch coming in. Over the next couple of minutes, we're going to be briefing each other on what we've seen. Our scientists will talk to the other scientists, telling them what we've collected, what's been used, what's available. That way when they continue the mission, they have a better understanding of what they have and, yeah, what's going on. So these don't have any polyps, or what do you call those antenna things?
They do actually, yeah.
Oh.
Those little bumps.
Oh, okay. So that's what they used to catch.
All right, so thank you everybody. We're shifting into our watch change now. We'll soon be reaching the summit. So
Yep, it was a pleasure.
it's a pleasure.
Thank you for spending your time with us. And yeah.
Do you want me to zoom out, sir? Or just come out?
So just kind of stand by as we make the change. It's been great. Have a great day wherever you are.
So here we are.
Yeah, I think I accidentally did. I was adjusting settings.
Do you think we can make 20 meters a minute again, Dan? Yeah? All right. The nominal rate.
The fun police is already calculating
Yeah, I am.
end of playtime.
Your tears bring me joy.
I'm sure.
Let's call it hour and 15 to surface for the whole deal. Well, that's assuming the top of the hill, which I think we'll be able to get to the top of the hill. Right now, I was advised to circle the drain. So, continue?
Continue.
All right.
Let's not get any shallower. Let's continue on this
Oh,
track
we're okay, just following contours. Roger.
for at least a little while. I mean, we got 3 hours.
Yeah.
And we only got, what, 20 or 30 meters to
Yeah.
get up.
This is a weird problem we found ourselves in.
It is. You cracked the whip a little too hard this morning.
I didn't. We didn't make it very far at all this morning.
We made it a lot further than I wanted to make it.
Megan did. Megan's mean. I don't know if you knew that.
Oh my gosh.
Yeah.
Well, welcome
All
to the 4-date, everyone.
I'm gonna put a ship move in.
Where you can tell we're into week 3 at sea.
Bridge nav. Let's do 30 meters 280. Yeah. Oh, hi, Flavio.
Let's welcome our viewers. It's 4 p.m. Palau time. And Brian, when you get a chance, you want to fill us in on what we're doing, what's the updates, where we're at.
Yeah, so we are currently exploring an unnamed, previously unexplored seamount about 65 miles west of Balbadob Island in Palau. We're at a depth of exactly 1,200 meters right now, which is about the shallowest we're going to get. We're going to pick up another 20 or 30 meters probably, so it'll be around 1150 when we recover. We started about 2,100 meters this morning and have been coming up this landslide scar, basically. We started on the top of the rubble pile and we've been working our way up until we got pretty close to the surface, and now we're spiraling around the feature. And we'll try and time the end of this dive with getting up to the summit with a couple minutes to look around. So we got a little less than 3 hours of exploration time left.
Did you say go wide?
Brian's happy to get on with it.
Yep, we can keep moving. And we're looking at this Prem node.
Thank you for that.
That is quite a lot of sea stars.
Yeah, they've been seeing them for a while. They seem to be very similar to the cookie star we collected, I think, on last leg. May passed down that they'd gotten several good looks at them. Can we look fuzzy coral bottom left?
Right.
It's off screen now, but not far. Oh, not coral, never mind. Cancel that request.
Right.
What type of sea stars are these?
Cookie stars.
Cookie stars.
I think they collected one on the last watch.
Oh, nice.
Or they were talking about cookies in the galley. I'm not sure. I wasn't paying attention.
It had something to do with getting cookies.
Something to do with cookies. Getting cookies. Yeah.
All right, so for this watch, let's just go ahead and do a roll call. You can just say hi when I call your name, say hi to the audience. We'll start with watch lead, Brian.
Hello, audience.
Science 2. Harold.
Hi, I'm Harold.
Data logger.
Hello, I'm Kinman Yao.
Our Hercules pilot, Daniel.
Hello from the Herc chair.
Atalanta pilot.
Hey everyone, I'm Jaden.
And our navigator.
Hey, I'm Chris.
Video. We didn't hear you, video.
Not an SPL, Amber.
Sorry. Hi, I'm Amber.
Hello.
Pushing too many buttons over here.
And lots of buttons you have.
Yeah. And I am Kim, sitting in the SciComm seat.
Why?
Can we zoom cup coral, please?
Sure. Go ahead, Amber. Push in.
Okay, push it in.
I don't know about cup coral.
Yep, I think I'm wrong again. Let me rephrase. Can we zoom an enemy?
Alright. If I can find a toe hold here. Push in a bit more if you want.
Copy.
All right, that is probably good. Thank you.
Copy.
Bridge nav, 60 meters, 295.
I forgot the technique there, Jaden, to move the ROV side to side instead of laterals. You can use the horizontals. You just got to, if you have forward in, and then you turn it. That's what I was trying to explain the other night, and I was botching it horribly. If you have forward in and then you twist, they don't reverse, they just go less forward. So if you twist just a little with forward in, then it won't blow it out.
Can we do a flyby on the big whip?
Yeah, for sure.
I just had a very funny realization. Can't believe I never thought of this until today, but the fact that they're called sea anemones just made me realize, wait a second, is there something on land that's just an anemone? I had never thought of that before, and it's a type of flower called an anemone.
I did not know that either.
I'm a